Thank you, and hello everyone, welcome back. This third session will be on understanding the impact of study design and the role of model-informed development of dosage selection and evaluation of new indications. My name is CJ Musante. I am the president of ISoP, also at Pfizer, and I am co-chairing this session with Stacy Shord from the FDA. So for this session, I will need to count 4 talks. The first will be by Dr. Retain. The second will be by Dr. Chunzi Li. The third will be by Dr. Li Zhu. And the third will be by Dr. Ying Oh. And I'll call each of them to the stage, and then we'll also be joined by panelists. At the end of the session, we'll have the Q&A. So for the first talk, it's our panelists. Beautiful. The first talk will be Design Considerations for Dosage Selection and Optimization for New Indications by Dr. Mark Retain from the University of Chicago. And please do look at the biographies of each of our esteemed speakers online. Thank you.
Thank you, CJ. I greatly appreciate the invitation to speak here today in this panel. I've never actually been to an ACOP meeting myself, but certainly read the pharmacometrics literature on a regular basis. So I'm pleased to just sort of provide a general perspective. And I think one way to think about this is you're looking at secondary indications for drugs. And now we're talking about drugs that are already approved for one indication. And the question is, what do we need to do as we seek dosing for subsequent indications? So there's 2 sets of issues to consider. One is, what do we know about the diseases, about the disease that the drug is already indicated for? And what do we know about the drug? And as far as disease, there's obviously tissue of origin in cancer. And, if the 2 tumors have some similarity, that's different than if, a drug's already approved for acute leukemia and now you think it's going to work in colorectal cancer. The goals of treatment. Are we trying to cure the disease? Are we trying to simply improve survival? Are we trying to simply improve quality of life? I mean, these are all issues to think about. Tumor burden is a potential factor to consider. And again, are we talking about patients with widely metastatic disease? Are we talking about the use of the drug in the adjuvant setting where we're treating disease we can't see? And then finally, at the molecular level, there can be differences in the molecular genetics. Even within a disease, within a stage, there can be major differences that influence how we develop the drug. And what do we know about the drug? Was the drug optimized as far as dosing and schedule? What do we know about exposure response, both for efficacy and toxicity? And so all of these are issues to consider. So I'm going to take you through some examples where, for the most part, they've been fairly illogical, and to sort of try and take you down the path of where I think we need to go. So let's start with this drug, rituximab, which is an anti-CD20 monoclonal antibody that's been around a long time. And if you read the prescribing information, you go, well, I'm not sure how we got to these different doses. Some are in milligrams, some are in milligrams per meter squared. It's kind of hard to understand why we need precision for some indications and we don't need precision for other indications. And in oncology, it's been, for example, CLL, non-Hodgkin's lymphoma, 375 milligrams per meter squared. That same dose is used in vasculitis and other rheumatological indications For example, rheumatoid arthritis, pemphigus, 1,000 milligrams. And so, so there's just not a lot of rhyme or reason when you read the prescribing information, and it's certainly not clear in the clinical pharmacology section of the label as to how we got to these doses. So a reasonable prescriber who wanted to understand the justification for the doses wouldn't be able to figure out either from the label or from whatever public data are available. Yet there, for example, are studies that could inform dosing. And here's a paper published in Blood that showed that high tumor burden was correlated with low AUC in large cell lymphoma. And in other words, the higher the tumor burden, the higher the clearance. Now, that's not all that surprising to see that relationship. Of course, you get greater target-mediated drug disposition. Patients with a high tumor burden are more cachectic and will have higher clearance. And it's certainly not clear that patients with high tumor burden should get a higher dose, but at least this generates some hypotheses that could be studied if one wanted to try and better rationalize the dose for this indication. And there's been an attempt to model using, what I think are reasonably robust modeling techniques, the pharmacokinetics of rituximab across disease states using a parallel first order and TMDD model. And you see, for example, that lymphoma, large cell lymphoma, has the highest volume. CLL has the lowest K10. And so, there's these efforts where you can, again, think about scientific approaches to these diseases that, haven't been applied either in the development or now that these data are available. In other words, what would you do with this? And if, and obviously it depends on how you feel about dose and exposure toxicity relationships as to whether you care about these potential population pharmacokinetic differences that could be exploited if one wanted to individualize dosing. Then we have nivolumab and a laundry list of indications. And here what we find is consistency. Consistency, for the most part, averaging 120 milligrams per week, 240 every 2, 360 every 3, 480 every 4. Original dose was 3 mg per kg. And so we have consistency. Again, in this case, there's no justification for the consistency. There have been attempts, again, population pharmacokinetics. Here's a paper published in CPT in 2019. And I think the most striking finding is that in melanoma patients, that the patients with the lowest clearance are adjuvant therapy for melanoma. In other words, they have a lower tumor burden, they have lower clearance, and the patients with advanced disease have higher clearance. And so again, we have these differences across disease states. And you see that a bit more here. Again, there's weight is certainly a covariate in the population analysis. Performance status is a covariate. Patients with poor performance status have higher clearance. Okay, so so you might say, oh, well, maybe that's why we need to give high doses to everybody because we're not very good at assessing performance status. That's not the right answer, and I'll tell you why in a second. But we have this kind of information. So, and the other issue is in looking at exposure-response relationships, one has to be concerned about confounding. So there's an inverse relationship between baseline nivolumab clearance and probability of response that cannot be overcome by increasing the dose. In other words, these patients with high clearance won't respond no matter what dose you give them. There's no evidence of greater efficacy of 10 versus 2, for example, or 10 versus 3 yet you have these patients with very high clearance that just won't respond on. That's because the high clearance is due to the cachectic state, increased protein turnover, including therapeutic monoclonal antibodies. And the green, for the sake of reading this graph, the green represents patients that benefited, at least responded, CRs and PRs. The red are patients that did not respond. And you can see that that in the group of patients with the highest clearance towards the top of that graph, there's a lot more dots in the red than in the green. And so this is a confounding. And so anytime you see an exposure response relationship, you have to question, is it real or is it not? And you will see an exposure response relationship for every monoclonal antibody in cancer, regardless of whether the drug is effective or not, just because the sickest patients will have the highest clearance and they will have the shortest life expectancy. And so you have to be very concerned about confounding. Furthermore, none of the doses of nivolumab are the right dose. They're all too high. And this is a BMS-sponsored trial published in Journal of Clinical Oncology in 2015, showing a completely flat dose-response relationship over more than a 30-fold dose range from 0.3 to 10 mg per kg every 3 weeks, yet the initial indication, initial dosing was 3 mg per kg every 2 weeks. And so 15 times high, the minimally effective dose is based on these data set. And there's other data that are consistent with this. In the phase 1 trials, responses were seen at 0.1 mg per kg. And so again, we don't have good logic to support the nivolumab labeling. Then turning to bevacizumab. Again, what you see is a progression of labeling, generally progressing towards the high end as time goes on. I mean, the initial approval was at 5 mg per kg. And then there was one study that suggested more may be better, even though the initial study suggested the more was not better. And then once it was figured out that, well, you can prescribe 15 mg per kg, physicians will prescribe 15 mg per kg, and 15 mg per kg generates 3 times as much in sales as 5 mg per kg. Why should we label in our new indications with anything less than 15 mg per kg? And so this has just gone on and continuing again without justification. Here's the initial trial, a well-done, randomized, dose-ranging phase 2 trial in metastatic colorectal cancer, chemotherapy alone, that's the control group, and then chemotherapy plus 5, chemotherapy plus 10 of bevacizumab, and 5 was selected since 10 was no better, 5 was better than 0, and that's how that dose was established, the right way. But then the commercial people took over, I guess. What can I say? And here's my current thinking on atezolizumab and bevacizumab dosing. It's not based on science, it's just based on revenue. And it may be that the Optimus of bevacizumab for some of its approved indications may actually be 0 rather than 15. So I think that there's a lot of work we can do on drug dosing. And unfortunately, FDA doesn't have the authority to require sponsors to do the studies that they are now requiring under Project Optimus for drugs not yet approved. And obviously, industry doesn't have any motivation, at least for their own drugs. Now, as I commented earlier, if one is developing a drug in combination with an already approved drug belonging to another company, there is a motivation to actually try and lower the dose of the other drug to try and reduce the total cost of the regimen that you're trying to get approved. All right, now here's an example of where I think it's been done right. Okay. I don't know if there's anybody from Novartis here, asciminib. And here there's mutation-specific dosing. And this is for chronic myeloid leukemia, Philadelphia positive. And there's the broader population for whom a dose of 80 milligrams daily is indicated, and then a subpopulation with a specific mutation with a 400 milligrams a day, 200 milligrams twice daily dosing recommendation. This is based on science. And it's based, if you go back to the original medicinal chemistry paper, you see the marked difference in the GI 50 or 90, I think it's a 50, between the wild type and the mutant. And so this led to a development plan to do mutation-specific dosing, and it was successful and justifiable. And here's some of the data. This is from the FDA review, exposure-response relationship for the two groups of subjects. Those that are wild type on the left, denoted as T315I minus, and those that have that mutant, T315I positive. You see the different exposure response profiles, and clearly a higher exposure is required for the T315I positive group. So preclinical data confirmed by clinical pharmacology and clinical data. All right. Well, we could go on for a while. Lenvatinib, we have a labeled dose of 8 to 24 milligrams. This is a multi-targeted kinase inhibitor. It looks like a lot of other kinase inhibitors. There's nothing really magical about this particular one, nor is there anything magically different about these different diseases and why they should be treated with different doses of lenvatinib, with the possible exception that maybe if you're targeting RET, you want a, maybe a lower dose because it's a pretty good RET inhibitor. The original dose was 24. And that was a dose that generally is not tolerated. It was, Eisai was really solely in charge of the drug at that time. And then subsequently, there's been much lower doses studied, going down to 8 milligrams in hepatocellular cancer for smaller patients. Then we have combinations in combination with pembrolizumab at higher doses. And again, it's obvious why it's in combination with pembrolizumab, because Merck acquired the license to co-market the drug. But again, the dosing has not been explored there to justify, in my opinion, 20 milligrams. We have co-labeling with everolimus. To some extent, that's based on toxicity concerns that you certainly can't give the 24 milligram dose. But this has made the commercialization of this drug very complicated because the two formulation strengths are 10 and 4. So selling doses between 8 and 24 milligrams requires them to come up with dose packs. And it's a different dose pack for each indication. And actually, excuse me, the most cost-effective way for a prescriber to use this drug is to prescribe 24 milligrams, regardless of what the dose is, because then the patient can actually, it's the same cost for whatever your dose is. So whether you're prescribing 8 milligrams daily or 24 milligrams daily, the monthly cost is the same. But prescriber really was interested in trying to conserve resources. They would split the packs. And I think the VA might actually even be doing that because I think they may have figured that out. All right, now I want to turn to a recent event. So Pirtobrutinib was approved in January of this year. Approved shortly thereafter, FDA released its what I call the Project Optimus draft guidance, the dosing draft guidance for oncology. And Pirtobrutinib is a BTK inhibitor. It's the Other approved BTK inhibitors are covalent binders. This is the first non-covalent reversible BTK inhibitor that was approved. And it was approved for an orphan disease, mantle cell lymphoma, approved at a dose of 200 milligrams daily and formulated as 50s and 100s. So you might say, well, it was approved right after Project Optimus draft guidance was released. Let's see what they did for dose optimization. And nothing. And basically, it was an accelerated approval, and there was no pre-marketing dose optimization. There's no post-marketing requirement for dose optimization. The FDA review included data for the relationship of starting dose and response rate. There was intrapatient dose escalation. So what was the dose at the time of response? The bottom line is you can't tell. You really can't tell. There's certainly no evidence that 200 milligrams is optimal. It might be optimal, just hasn't been proven. May also be the 100's optimal. And certainly 50 has some activity. So, and again, drug has toxicity. And so the question is, well, what could be done? And here's, for example, the FDA's attempt to analyze the relationship between exposure response and dose response. The problem was there's just very little data at a lower dose. And they note that there was a trend of lower response rate at a 50 milligram dose of 29% compared to 61% at 200 milligrams, okay? Maybe that's true, maybe that isn't. The problem is that the modeling is probably flawed, it's probably confounded, the exposure response data, because patients with decreased albumin who generally have slower metabolism, slower clearance of small molecules. In this case, they had higher clearance, okay? That tells you that there's a problem, that perhaps this relates to protein binding, that the patients with the lower albumin have less bound drug and therefore less total drug. And so this, again, without randomized dose ranging data, the exposure response relationship is uninterpretable and certainly shouldn't be used to waive any requirement for post-marketing dose optimization. Furthermore, it's not just an orphan drug. It was, while mantle cell lymphoma has orphan disease designation, Eli Lilly is developing the drug more broadly. They have multiple trials running in chronic lymphocytic leukemia. And so here's an example of a trial design that was disclosed during this year's ASCO meeting, comparing pirtobrutinib with ibrutinib. And you see that it's either treatment naive or previously treated, randomized 1-to-1, 200 milligrams pirtobrutinib or 420 milligrams of ibrutinib. This is an opportunity for dose optimization that was not required, and certainly there are abundant patients with CLL that could be randomized to really learn about the dose of this agent. And in fact, they're running 4 phase 3 trials, all at this same dose. Presumably, they're all 2-arm trials, and there is conceivable that one of the or more of these trials is using a lower dose, but I've not heard that, and I've not seen any evidence to support that. So this is a missed opportunity despite the Project Optimus guidance, and it goes right to the heart of this session. What do you do with second indications? And here we have a drug, wasn't optimized for the initial approval, now being developed for much broader indications. If FDA is serious about Project Optimus, it can't let sponsors take this route to avoid dose optimization. All right, so where do we go from here? There's no prior dose optimization. It basically, randomized dose ranging trials could proceed similar to drugs being evaluated for their initial indication. And this may include doses both lower and higher than the already approved dose. So in other words, it's not really relevant that the drug's already approved. If it's never been optimized, optimize it. Now, and the trickier and the real art comes in, and this is both art and science. What do you do when you've got clear dose optimization for the initial indication? Everything's done right, and you really understand the drug. So you can start off with the hypothesis, well, it's going to be the same dose, and then think of reasons why that hypothesis could be wrong, and then decide whether it's worth investing to test those hypotheses. And so think about disease type, disease stage, patient characteristics that affect drug pharmacokinetics or pharmacodynamics, and the risk-benefit ratio threshold. So I just have one more example to talk about. Rubraca, I forget what it is. Anyway, it's a PARP inhibitor. And rucaparib, I think. And so it's a PARP inhibitor that was originally approved in ovarian cancer as a maintenance therapy. And basically, though, you know, there's some evidence that you know, about exposure response. And, you know, the problem here is that this drug is toxic and there's exposure toxicity relationships, and certainly patients with decreased albumin or other poor performance status characteristics are at greater risk for toxicity. And so there are many potential reasons why the optimal dose for this drug may vary by disease setting. So first indication was maintenance therapy of ovarian cancer after major response. Then they got a second indication, recurrent BRCA mutant metastatic prostate cancer. Very different disease settings. So in the first group, the original approval, you're worried about neutropenia because these patients just got aggressive chemotherapy for ovarian cancer, and you're now putting them on maintenance therapy. They're going to have decreased marrow reserve. In this prostate cancer group, they're often going to have a decreased performance status, and they're going to have decreased albumin. They're going to have decreased renal function. And all of these things are going to lead to decreased clearance of the drug in this patient population. Not to mention, the drug was essentially developed only in women, and now you're turning, you know, testing it in men. And so, yes, you'll understand the safety of that particular dose, but you won't understand the dose range, and there's only so much you can extrapolate from the prior data. So I think we have plenty of work to do. This is all an effort in progress, but I think everybody needs to do their part. I think FDA has to stand by Project Optimus and continue to support the notion of dose optimization. And I think sponsors need to continue to think carefully about the many challenges here. So thank you. I look forward to the discussion.
Thank you, Dr. Retain. I think that really sets the stage for the session well. So now I'd like to welcome to the stage our next speaker. This will be Dr. Chunzi Li. She'll be speaking about model-informed dose optimization for antibody drug conjugates and T-cell dependent bispecifics across indications.
Thank you, CJ, for the introduction. So first, I would like to thank you, the meeting organizer, to the kind invitation. So good afternoon. So it is my great pleasure to share with you model-informed dose optimization for antibody drug conjugate and the T-cell dependent bispecifics across indication. So first, I just want to, here's the outline of my presentation. So first, I would like to give you an overview of ADC and the TDB and their associated dose optimization challenges. And then I would like to share with you the dose optimization considerations for ADC and the T-cell dependent bispecific across indication using Enhertu and leukemia as a case example. And lastly is the summary. So here is the mechanism action of antibody drug conjugates. So it undergo target-mediated endocytosis to lead to the release of the cytotoxic payload inside the cells, and therefore lead to the tumor cell killing and the potentially adjacent tumor cells. So one of the development challenges for ADC has a relative narrow therapeutic window. In comparison, T-cell dependent bispecific is another important class of molecule for the cancer treatment. So here are showing the mechanism action of the T-cell engager. So basically, the T-cell engager bound to the CD3 cells on the CD3 receptor on the T cells and the targeted antigen on the tumor cells to form the immune synapse, leading to the release of the cytokine and also granzyme B perforin to kill the tumor cells. So one of the biggest challenge for the dosage related to the TDB in general is the CRS, on-target acute toxicities. So usually occur acutely following the first dose and dissipate over the time. So to date, there's 11 ADC and 8 TDBs has approved by FDA. And with most of the T-cell engagers was approved for the last 2 years. So in today's presentation, I will mainly focus on Enhertu and Lusimil as a case example to show how we do dose optimization across different indications. So first on ADC, how we select the dose for first indication. Given ADC have relative narrow therapeutic window, actually there's a multiple diverse novel approach has been used to risk mitigate safety while maintain the efficacies. So due to limited time, I won't go through the detailed approach. So we do have detailed description of these different approaches and associated consideration published in the CPT 2021. So one thing I would like to highlight from this slide is the last approach is randomized dose finding study become increasingly important for the ADC to find appropriate dosing and scheduling, especially given the newer generation of the ADC usually have wider therapeutic window, so we can observe efficacy and safety across multiple doses. So how about for the subsequent indication for the ADCs? Among the 11 approved ADC, actually 5 of them only have 1 indication approved. And we do have 6 ADC with more than 1 indication was approved, with a total 8 new indications, subsequent new indication. So among these 8 new subsequent indication, 3 of them using the same dosing schedule, and 4 of them using the similar dosing schedule with some minor tweak. And we only have 1 situation using different dosing schedules. So here are the detailed list of the ADC and the subsequent new indication and using same or similar dosing schedules. As you can see here, when we move from later line to early line indications, there are increasing trend to cap the dose treatment durations. So which is, instead of treat up to progression for the later line disease. So which is likely, like Mark mentioned, is due to the higher safety and tolerability bar for the earlier disease. Additionally, we can also change in the scheduling to match the standard of care scheduling to provide additional patient convenience. And we only have 1 situation using different dosing schedule, and which is Enhertu, trastuzumab deruxtecan. And when they change from MBC to advanced gastric cancer, the dose was changed from 5.4 to 6.4 mg per kg. So overall, based on this analysis, first acknowledge the data is quite limited, but from this approved ADC, you can see majority ADC using the same or similar schedules with 1 exceptions. Well, sorry, I have a computer I try to. So in today's presentation, I will share with you the case example for trastuzumab Enhertu for the first indication and the subsequent indication dose selections. So first is the dose selection for Enhertu for the clinical dose selection for the first indication, which is HER2 positive MBC. So there are two things I would like to highlight in this dose finding strategy. So first is robust dose finding is critical to identify Enhertu clinical dose. So as I show here, the schema, there's two studies to support dose selection for Enhertu. So the phase 1 first in human dose escalation, as you can see, they explore a very broad range of dose from 0.8 to 8 mg per kg. And it is noted MTD was not achieved. And based on the safety efficacy, 5.4 and 6.4 mg per kg was selected to further expand in the dose expansion. Additionally, a randomized dose finding phase 2 study was also conducted between these two dose levels. And based on the randomization dose studies, along with the phase 1 study, there's one dose was selected, continue expand up to 100 patients. So together, the results from phase 1 and phase 2 study ultimately was used for the accelerated approval of Enhertu. And the other points I would like to highlight is integrated quantitative analysis, the importance. So in order to support RP2D selection for Enhertu, ER analysis, spore response analysis was conducted, showing here there's a positive ER relationship for both efficacy and safety. And the model also predict the different efficacy endpoint and safety endpoint between the two dose levels. As you can see, consistent with the positive ER relationship, the higher the dose, which is 6.4 mg per kg, tend to have better efficacy, but however, also have worse safeties. So due to the safety concern of the interstitial lung disease, 5.4 mg per kg was selected for the HER2 positive breast cancer. So how about for gastric cancer? So, I think it is known that monoclonal antibody, like Mark mentioned, that tend to have faster clearance and lower exposure in advanced gastric cancer as compared to other solid tumor is likely due to cancer cachexia, sickness disease. Additionally, maybe also proteolytic degradation may also, gastric protein leakage also potentially play a role. And there's several molecules already observed such a phenomenon, such as Herceptin, Perjeta, Casella, and Avastin, etc. So given this prior analysis, the exposure of Enhertu was compared between GC and the BC. So as you can see, the upper panel, left panel of the box plot, the steady state AUC of the ADC conjugate was lower in the gastric cancer patient as compared to the breast cancer patient. They also compared the GC 6.4 versus the BC 5.4 dose. Again, when you change the dose, their exposure are overall comparable. But however, PK difference does not always translate. You need a different dose. We need to put the PK changes in the context of efficacy and safety. So as a result, the sponsor conduct exposure response analysis for both safety and efficacy. Again, here you can see, consistent with as MBC, we see a positive ER relationship for both efficacy and safety. And again, based on this modeling, we predict that 6.4 mg per kg in gastric cancer will have better efficacy compared to 5.4 mg per kg. From safety perspective, and they also predict GC at 6.4 mg per kg, at 6.4 mg per kg will have comparable safety profiles as breast cancer at 5.4 mg per kg, which is in line, the exposure, they are overall similar. So together, based on this analysis, 6.4 mg per kg was selected as the clinical dose for HER2 positive gastric cancer. So now I want to switch gear to talk about T-cell dependent bispecific, using leucemia as one of the examples. Mosun is an anti-CD20, CD3 bispecific antibody for the treatment of B-cell malignancies. So here's the mechanism action of Mosun. So basically, Mosun brings T cells to the tumor cells, form immune synapse, lead to the release of cytokine, granzyme B and perforin, ultimately kill the tumor cells. So the key development challenge for Mosun, first, as I mentioned earlier, is the CRS. which is on-target acute toxicities, and which occur acutely following the first dose and gradually dissipate over the time. Another challenge in the dose finding is complex target engagement. So there's no simple PK target based on preclinical studies. And then lastly, phase 1 dose response relationship are all may potentially confounded by the patient prior anti-CD7, anti-CD20 therapies. So first, I think we want to risk mitigate CRS. In order to risk mitigate CRS, we need to understand IL-6 and its kinetics. Because we know patients with CRS tend to have elevated IL-6 levels. And so therefore, IL-6 is an important biomarker for CRS to get the inference of CRS risk. So we developed a QSP model to describe the IL-6 kinetics. So the QSP model captured kinetics and the cell trafficking of the both T cells and the B cells, and across physiological compartment, which is including blood, bone marrow, lymph node, spleen, and also tumor. Additionally, the QSP model also captured Mosun pharmacology, namely from the immune synapse and leading to the T cell activation and the IL-6 release. So we use this model to predict different dosing scheduling, how that impact on the IL-6 kinetics. So here just showing the two different doses as an illustration perspective. As you can see, the model predict if you add a smaller dose using 1.6 as an example, and before the target dose, we will have much lower IL-6 levels, the peak levels, as compared if we directly use target dose, which is we call fixed dosing. So based on this simulation, we gain the insight. So the QSP model actually help us to guide phase 1 dose escalation strategies. So instead of to have one dose escalation cohort, we strategically planning 2 dose escalation cohorts. So we will start with the dose escalation with a fixed dosing schedule. Once we start to see low-grade CRS and the IL-6 release, we start to trigger the second dose escalation with the step-up dosing. So here are showing the observed IL-6 profile for step-up dosing. As you can see, the profiles are pretty much aligned with the QSP predictions. Sorry, okay. So now I want to switch gear to talk about how we use model to characterize the key driver for clinical response. So the graph on the left, you can see this is a dose response relationship for step-up dosing. And the orange line is the CR complete response, and the purple line is objective response. So you can see there's no clear dose response relationship across different doses. It is, the reason is still not fully unknown. It's maybe due to small sample size or maybe confounded. So one of the confounding factor we found out is due to prior anti-CD20 therapy, which means the residual rituximab can compete with the Mosun on the CD20 binding. So lead to the less formation of the immune synapse and therefore less anti-tumor activities. So I think in order to correct that, potential confounding factor from rituximab, the competition, we calculate RO based off the Mosun to CD20 by accounting the binding constants and also the circulating concentration of the both molecules. So when we take account these, confounding factors, as you can see, when we use RO as explorer metrics to look into explorer response relationship, we can see much clear explorer response relationship for both OR and the CR. So together, I think I can illustrate to you that the model actually help us to model-based dose strategy actually help us expand the therapeutic index for the molecule. And based on this modeling, we were able to justify 1 to 60, 30 mg regimen in the relapsed refractory follicular lymphoma, which lead to the ultimate accelerated approval of this molecule in 2022. So next, I want to discuss a little bit, how about for the additional indication? In the subset, in this case, another B-cell malignancy, CLL. So in order to answer this question, what would be the dose for CLL, we first need to compare the disease similarities and difference. As you can see here, compared to NHL, CLL is a bone marrow disease, have much more tumor B cells in the circulation. So in order to confirm this disease understanding, we also leverage our historical clinical trial data to compare the baseline B cell distribution between relapsed refractory NHL versus CLL. As you can see clearly, CLL have much higher baseline B cell count as compared to NHL. Additionally, per MOA, we also look at the T cell distribution. Overall, they are pretty much similar compared to two disease. So then we leverage the QSP model we have been developed to guide dose finding strategy for CLL by taking account the high disease burden we have been known. So the first question from the team is, what kind of dose finding strategy we should use? Should we do a simple safety run-in? Should we do standalone dose escalation? Given we based on the QSP model and also the understanding of the CRS biology, we know the higher disease burden in the circulation, the higher the risk of CRS. So clearly, we could not do simple safety run-in in this case. So we have to do standalone dose escalation. And additionally, the model also help us to predict what will be the starting dose for this disease to achieve target dose in 2 cycles to make sure this patient can benefit these drugs while risk mitigate CRS risk. So we want to make sure the predict circulating IL-6 is within the range observed in the NHL. On the other hand, we also want to make sure there's a continued B cell killings in the circulation in the first 2 cycles to make sure the patient can gain some benefit from these studies. So this study is still ongoing. So hopefully in the future, we will come back to share with you the story for the selection. So lastly is the summary. Dose optimization strategy is not one-time thing. Should be seamlessly integrated early into the clinical development plan and also cross life cycle planning. In general, we can call that dosing CDP. So you need to plan early to gather preclinical evidence across indications and also including patient of interest in the first in-human studies. Dose optimization in the subsequent indication should be science and data-driven and fit for purpose. It is not scalable to do full-blown dose optimization in every indication, every combination. As I'm showing for the ADC situation, although the data is limited, the same or similar dosing schedule has been used for ADC for the subsequent indications, which is likely due to the similar pharmacology of the ADC across different indications. But on the other hand, we also should not do another extreme. We assume the same dose will automatically apply to other subsequent indications. Just like Mark mentioned, we need to think about what are the rationale. So different dosing schedule may be needed when there's a difference in tumor and disease biology, like I illustrated for Mosun in CLL and NHL. Additionally, you can also think about the patient population. Earlier disease, they may have different benefit risk profiles and also different requirement for the safety threshold. And then lastly, PK/PD. If we do aware some PK difference across different indication, we need to put that into the consideration. But I also want to emphasize, PK difference does not always warranty a different dose. You need to put the PK difference in the context of safety and efficacy. Lastly, I want to emphasize quantitative pharmacology and the mechanism modeling play an integral role in the clinical development. So as an example I'm showing here, can guide those findings, study design and strategy, can help us identify appropriate dose and the schedulings, and also can help us accelerate the development and also enhance the success of ADC and TDB development. So with that, I would like to make an acknowledgement my Genentech Clean Farm colleagues, Amita Joshi and her great support, and also Roche Oncology Dose Optimization Working Group, Mosun project teams, and the patient contribute for the data. Thank you for your attention.
Thanks very much, Chunzi. I think that nicely characterized some of the considerations for ADCs and TDBs when thinking about dosage optimization. Our next speaker will be Dr. Li Zhu, who will be talking about model-informed dosage optimization strategies for immune checkpoint inhibitors, multiple indications, same dosing regimen.
Thanks, CJ, for the introduction, and thanks the organizer for the opportunity to speak. So immune checkpoint inhibitors revolutionized cancer therapy and reinvigorated immuno-oncology. Immune checkpoints are receptors expressed on the surface of immune cells that can exert either inhibitory or stimulatory effect on the immune system. Of the many immune checkpoints identified, 3 had approved therapy. CTLA-4 inhibitor Ipilimumab was the first immune checkpoint inhibitors approved in 2011. PD-1 inhibitors Pembrolizumab and Nivolumab was approved in 2014, and last year saw the approval of LAG-3 inhibitor Relatlimab used in the fixed dose combination with Nivolumab. One of the key differences between immune checkpoint inhibitor and other cancer therapies, such as chemotherapy, TKIs, are that immune checkpoint inhibitors does not kill cancer cells itself, but rather it primes the patient's immune system to combat the disease. And as such, there is a biological rationale to potentially develop immune checkpoint inhibitors, not just against one cancer, but multiple indications. I think Dr. Shah also cited this dose optimization draft guidance as a follow-up to FDA's Project Optimus, in that it stated that different dosages may be needed, may be required for different disease due to the potential difference in tumor biology, patient population, treatment settings, etc. And a strong justification is required for the choice of the dose before a registration trial can be started for subsequent treatments. And as we mentioned, immune checkpoint inhibitors are likely to have a development paradigm that's geared toward multiple indications. It is important for us to understand the considerations for optimal dosage selection to facilitate such a development paradigm. So the illustration on the right should really be very familiar with this audience. I actually thought no Project Optimus talk should be without it. Clearly, it shows a plateauing dose efficacy relationship, but a somewhat steeper dose toxicity relationship. And the interplay between the two relationships argues for optimal dose rather than a maximum tolerated dose. But one can also visualize a somewhat shallower dose toxicity profile, whereby creating a wide therapeutic window that can potentially enable a selection of an optimal dose that's both safe and efficacious, but can also accommodate the differences in tumor biology, in patient population, in treatment settings, etc. Obviously, on the other end of the spectrum, we can also visualize a much steeper dose toxicity relationship, which will require a more individualized dose selection based on the characteristics of a particular patient population or disease. So we'll look at the examples from all 3 classes of immune checkpoint inhibitors and see how their exposure response profile fits into the scenarios described here, and how their consideration influence their development for multiple indications. So of all the immune checkpoint inhibitors, PD-1, PD-L1 inhibitors have the most approval and most clinical experience. A PD-1 inhibitor such as Nivolumab works by binding to PD-1 expressed on the T cells and blocking its interaction with PD-L1, PD-L2 expressed on the surface of cancer cells. And as such, reinvigorating the exhausted T cells, unleashing the cytotoxic cascade and result in tumor cells destruction. So nivolumab as a PD-1 inhibitor was approved in 2014, initially in advanced melanoma, and since has been approved in an extensive and diverse set of tumor types. And of all the approvals for nivolumab as a single agent or in combination with chemotherapy or TKI, one clinically equivalent dose was approved. If you are familiar with this area, you know that nivolumab was originally approved as a weight-based dosing 3 milligram per kilogram every 2 weeks, which has since been updated to a more convenient 240 milligram every 2 week flat dosing, or 480 milligram every 4 week, or 360 milligram every 3 weeks. I consider these different dosages to be therapeutically equivalent, and very similar examples can also be found in other PD-1, PD-L1 inhibitors, such as pembrolizumab, atezolizumab, durvalumab, where one clinically equivalent dosage was approved for multiple indications. I think Dr. Retain, in his earlier remarks, discussed the nivolumab dose, whether a lower dose should be approved in general or at least for some tumor indications. I will use the next few slides to describe the clinical development of nivolumab, particularly some of the early clinical development strategies, the model-informed integrated analysis, and really putting it all together, the totality of the data, not just the clinical data, but also the drug development considerations, as well as patient access considerations that led us to select the dose that was eventually approved. Given PD-1's potential to be developed for multiple indications, right after the SAD and MAD in a mixed tumor type, cohort expansion focused in 3 different tumor types. They are melanoma, NSCLC, and RCC. As you can see, extensive dose finding was conducted in all 3 tumor types. The dose range is fairly wide, from 0.1 milligram to 10 milligram per kilogram, and there are decent sample size per dose cohort. Additionally, there were also 2 randomized phase 2 trials for RCC, again with extensive dose ranging, which provided data to inform efficacy and safety, as well as generating data for biomarkers. And based on the rich data set generated from these dose finding exercise, a model-informed dose optimization dose selection was conducted based on the totality of PK, target engagement, safety, and efficacy data. And the 3 milligram per kilogram every 2 week dose was selected for all 3 tumor types for their pivotal studies, based on which positive benefit and risk was demonstrated. And the 3 milligram per kilogram dose was approved in melanoma in 2014 and in NSCLC and RCC in 2015. So nivolumab has linear PK across the dose ranges evaluated from 0.1 milligram to actually 20 milligram per kilogram, as you can see in the panel on the left. Nivolumab PK is also similar across the different tumor types initially evaluated, as you can see in the panel in the middle, as well as in the forest plot on the right, which is based on a comprehensive population PK analysis. Although subsequent clinical development indicated in some tumor types, such as adjuvant treatment of melanoma, nivolumab clearance is different. And in some liquid tumors, such as classical Hodgkin's lymphoma, again, nivolumab clearance is different. However, based on the exposure response characteristics of nivolumab, which we'll discuss on the next slide, we don't consider these clearance differences to be clinically relevant. So the left panels shows the dose or exposure efficacy relationship by each tumor type for nivolumab. As you can see, the exposure efficacy relationship is fairly flat for melanoma. That's one on the leftmost panel, bottom panel. For RCC, there's a slightly upward trend for exposure response, but if you look at dose response. As Dr. Retain also pointed out in his earlier slides, there's no dose response for RCC. For NSCLC, however, there is a marked dose response or exposure response. Subject treated with the lower dose, 1 milligram per kilogram, have lower response rate compared to subject treated at 3 milligram and 10 milligram per kilogram. On the right-hand panel shows the safety by dose levels with all three tumor types combined. Again, here the top panel is grade 3 plus TRAE. The bottom panel is adverse event leading to discontinuation. Here you can see similarly a relatively flat safety dose safety relationship is observed for nivolumab, especially for the dose level from 1 milligram to 10 milligram per kilogram. And importantly, nivolumab has been shown to be safe and tolerable for the high dose of 10 milligram per kilogram in a fairly large number of subjects across different tumor indications. Nivolumab peripheral receptor occupancy was initially investigated in one of the melanoma cohort expansion portion of the study, and dose-hour relationship was found to be plateauing at the low dose of 0.3 milligram per kilogram. We also got longitudinal receptor occupancy data based on one of the randomized phase 2 study in RCC. Again, the peripheral receptor occupancy was saturated at the low dose of 0.3 milligram per kilogram and actually maintained throughout the 3-week dosing interval. However, we do know that peripheral receptor occupancy did not differentiate between the doses, did not differentiate between different tumor types, and we consider the peripheral receptor occupancy to be informative of the minimum effective dose, but less valuable in actually selecting an optimal dose. As we all know that the receptor occupancy at the site of action, in this case is tumor, could be very different from that in the blood. So we used a mechanistic PK model to predict the tumor receptor occupancy. So I wouldn't go into details of the mechanistic model, but the model, as you would imagine, indicated that to saturate tumor receptor occupancy, a much higher dose or much higher exposure is needed. But the advantage of using a mechanistic model is it can help us visualize different disease factors, tumor biologies, and their impact on the exposure-hour relationship. So here, we actually investigated the impact of target expression on exposure-hour relationship. Here, the blue line and the black line are high and low PD-1 expression. The solid line and the dotted line represent the low and high PD-L1 expression in the tumor. So clearly, you can see different tumor biology, different target expression could require a different dose. So now we actually have a lot of information, right? So we have done extensive dose ranging across multiple tumor types. We know nivolumab clearance is similar across different tumor types. The PK is linear. It has a wide therapeutic window. The exposure response relationship for efficacy and safety are fairly flat. And we know the peripheral receptor occupancy is saturated at low dose, and the tumor receptor occupancy indicated that different tumor biology, different disease could require a different dose. So how do we put all this information together in selecting optimal dose? We could certainly individualize the dose selection for each subsequent tumor type. But by doing so, it will greatly delay the development of subsequent indications and consequently patient access to these treatments. And inevitably, I would imagine will leave a larger number of patients with a subtherapeutic dose simply because of inter-individual variability. Or should we actually, in this case, take advantage of the wide therapeutic window and the safety margin afforded by the safe and tolerable high dose of 10 milligram per kilogram to select an optimal dose that has the favorable benefit and risk that can also accommodate the heterogeneity in the different diseases. So the selection of 3 milligram per kilogram dose is based on this latter principle. And the benefit and risk has been demonstrated over and over again in subsequent indications in their pivotal trials. And importantly, this has greatly facilitated patient access in a very timely manner. So you would imagine more than a dozen subsequent indications were approved in less than 9 years' time since the initial approval in 2014. And additionally, the wide therapeutic window, as well as the rationally selected 3 milligram per kilogram dose, also facilitated the more informed pathology update to the more convenient every 2-week dosing and every 4-week dosing. So let's move on to CTLA-4 inhibitors using Ipilimumab as an example. Ipi works by blocking the binding between CTLA-4, which is expressed on T cells, and CD80-86 expressed on antigen-presenting cells. Ipi has the function of augmenting the activation, proliferation of T cells in both the tumor-draining lymph node as well as in the tumor microenvironment. Ipilimumab could also have impact on the T regulatory cells, reducing Tregs activity. So Ipilimumab was originally approved in 2011 in advanced melanoma at a dose of 3 milligram per kilogram every 2 weeks. Subsequently, Ipi was also approved as a monotherapy in the adjuvant treatment of melanoma at a dose of 10 milligram per kilogram every 2 weeks for 4 doses. When Ipi was used in combination with Nivolumab, it has also been approved in quite a number of different tumor types, however, with different dose and regimens, as you can see in the color coding in the table. A similar story might be developing for Tremelimumab, which is another CTLA-4 inhibitor. As you can see, very different dosages has been approved for HCC and SCLC when the drug is used in combination with Durvalumab. So Ipi as a monotherapy, the clinical development focused on melanoma. So right after the initial dose escalation, dose finding was carried out in the form of 2 randomized phase 2 trials in previously untreated and previously treated melanoma. Again, you can see there's extensive dose ranging from 0.3 milligram to 10 milligram per kilogram. And these phase 2 data informed us that Ipi has a narrower therapeutic window. Higher Ipi dose is correlated with better efficacy, however, with more toxicity. So balancing the two, we selected 3 milligram per kilogram dose for the advanced melanoma for its pivotal trial. And this dose was subsequently approved based on significant OS benefit, but not without a post-marketing requirement to conduct a dose optimization trial to see whether 10 milligram per kilogram dose is in fact a better dose. So that trial has been run and has since read out and actually confirmed the benefit and risk with the 3 milligram per kilogram dose. So for adjuvant treatment of melanoma, the 10 milligram per kilogram dose was actually selected for its pivotal trial, apparently in an attempt to maximize the efficacy. So that dose was also approved, and a post-marketing dose optimization trial was also run. This time was run as an ISR, investigator-led trial, which actually indicated that the 3 milligram per kilogram dose is a better dose, has a better benefit and risk, being no less active, but less toxic. So here's the exposure response analysis conducted for the Ipi monotherapy. Again, Ipi has a positive exposure response relationship for safety and efficacy. Higher Ipi exposure, higher Ipi exposure do have a better response rate, longer overall survival. However, it does have greater grade 2 and grade 3 IRAEs. So you can see this is a very different scenario between Ipilimumab and Nivolumab, which have a wider therapeutic window that allowed us to select one optimal dose for multiple indications. For Ipi, it has a narrower therapeutic window. So especially the steep dose toxicity profile really required a much more robust clinical and modeling effort to really identify an optimal dose for both the initial indication and subsequent indication, as changes in the disease setting in the patient population could significantly change the assessment of benefit and risk. So if we think it's an extraordinary effort to actually finally nail down the optimal dose for Ipi monotherapy, as you can see, it takes a randomized phase 2 trial, a pivotal trial, and a post-marketing dose optimization trial. It is even more complex when it comes to Ipi and Nivo combination. So the two immune checkpoint inhibitors have complementary MOA, and early clinical evidence suggests there's better efficacy compared to either agent dosed alone. However, there's also combination toxicity. So I actually wouldn't go into details of this table, but just to show you the extent, right, the extensive dose finding that was conducted for each and every different tumor type. And this goes beyond the initial approval of the combination in melanoma. Very early on, we realized that by simply combining the two agents at its monotherapy approved dose, it's not going to be tolerable. Different disease have a different dependency on Ipi for efficacy, which will then have to be balanced against the combination toxicity. For the NSCLC, for example, this indication seems to be particularly sensitive to the combination toxicity. However, this indication also seems to be very responsive to low-dose Ipi. So extensive dose ranging finally led to a very different combination dose for Ipi, which is 1 milligram per kilogram every 6 weeks, treating continuously, compared to the monotherapy approved dose, which is 3 milligram per kilogram every 3 weeks for 4 doses, sorry. I know I'm running over time. And okay. So last example is Relatlimab, which is a LAG-3 inhibitor used in the fixed dose combination with Nivolumab. The fixed dose combination product is called Opdualag. Very similarly, Relatlimab works by blocking the binding between LAG-3 and its natural ligand, MHC class 2 and soluble FGFR1. Relatlimab currently is only approved in melanoma. However, there are several clinical trials that's ongoing to evaluate the therapeutic potentials in other indications. So Relatlimab clinical development started with extensive dose ranging in the dose escalation, particularly focused on combination with Nivolumab. And these early clinical data indicated Rela-Nivo combination is actually well tolerated and nothing like the Ipi-Nivo combination. So the cohort expansion focused initially on signal seeking in multiple indications, as well as a confirmatory efficacy in post-IO melanoma. And this also included a assessment of regimen, being Rela 80 milligram every 2 weeks versus 160 milligram every 4 weeks in combination with Nivolumab as the corresponding dose. So a formal dose optimization was also run to evaluate a low-dose Rela, 160 milligram every 4 weeks versus a high-dose Rela, 480 milligram every 4 weeks, while keeping Nivolumab dose the same as 480 milligram every 4 weeks. And this dose optimization trial was run in a randomized fashion. So the low-dose Rela regimen, 160 milligram every 4 weeks plus Nivo 480 milligram every 4 weeks, has shown to have the positive benefit and risk in the pivotal trial, and this dose was eventually approved. So again, if we look at the exposure-response relationship, we understand that the combination significantly reduced the risk of progression or death, but the improvement in efficacy was actually similar across the Rela dose evaluated, being 80 milligram every 2 weeks, 160 milligram every 4 weeks, and 480 milligram every 4 weeks. And similarly, the combination also significantly increased the risk of grade 3 IMAE, grade 2 IMAE, grade 3 drug-related AEs compared to nivolumab monotherapy. However, the risk was not increased from Rela 160 milligram to 480 milligram. We also look at the objective response as a clinical endpoint, which is shown on the bottom left figure. So here you actually see a positive exposure OR relationship. However, objective response as a clinical endpoint lacks the duration of response component, and we do know in melanoma, PFS was dose independent. So these dose finding results, as well as the exposure response analysis results, supported the 160 milligram dose for melanoma. So Rela seems to have fairly flat exposure response characteristics. It's absolutely nothing like Ipi, it's more like nivolumab. However, is the currently approved Rela dose 160 milligram Q4 is going to be broadly applicable or broadly optimal for subsequent indications. There are a few considerations. First, unlike nivolumab, which has linear PK, Relatlimab actually have nonlinear PK due to the presence of soluble target as well as faster internalization rate. So you can very well imagine a higher dose may be needed to fully saturate the nonlinear portion of the clearance in a tumor type that perhaps have more tumor, have higher tumor burden, have higher target expression as well. And again, we used the mechanistic model to project Rela receptor occupancy. Here, we show a worst case scenario, which included scenarios like low tumor vasculature volume, low tumor drug penetration, high target expression in the tumor, and etc. So in such a worst case scenario, as you can see, we will predict we need a higher dose, such as 480 milligram dose to fully saturate the tumor receptor occupancy. And similarly, we did see the trend of a positive exposure OR relationship. This didn't translate into PFS differences. However, there's uncertainty in a less immunogenic tumor or less responsive tumor. And most importantly, these dose finding exercises supported the safety of the combination, and the flat dose safety relationship allowed us the opportunity to further explore Rela dose. So I think for Rela, the jury is still out. So in summary, the immune checkpoint inhibitors have demonstrated breakthrough treatment effect by boosting the tumor-specific immune response in a wide range of cancer types. The ability to identify a harmonized dose across tumor types or line of therapy greatly expedite life cycle development following the initial approval of the drug. And it's important for us to generate preclinical information for pharmacodynamic and target engagement to help us set the stage for dose ranging, and early clinical studies incorporating dose finding in multiple tumor types is critical in generating the data to inform the integrated PK/PD analysis, the exposure response analysis to help decide on the RP3D. And critically, optimal dose selection should consider the shape of the dose response, exposure response, as well as the uncertainty heterogeneity inherent in the multiple indication. Thank you. And just to, just to acknowledge patients who participated in our trials, as well as clinical development team at Bristol Myers Squibb, as well as my clinical pharmacology, pharmacometrics colleagues, past and present at Bristol Myers Squibb. Thank you again.
Thanks very much, Dr. Zhu, for those very nice contrasting examples of immune checkpoint inhibitors. So our next speaker is Dr. Ying Oh, who will be discussing model-informed dosage optimization strategies for targeted oral agents, different dosages, different indications.
Good afternoon, everybody. Thank you, and thank you, CJ and the FDA and the organizer for inviting me for this very thought-provoking workshop. It certainly is my honor to share with you all additional case studies and stories to wrap up this workshop. And I'm going to go on to the next slide, please. Since the approval of Gleevec almost 22 years ago, oral targeted agent continue to play an important role in the cancer treatment. And one unique feature of the oral target therapy is its ability for patients to take it at home either once per day or twice per day. So the convenience is really what counts. Now, just like many speakers that have shared earlier, the totality evidence approach can be used to bridge from one dose to a different dose level or to different indication. Now, with that in mind, today I will begin by addressing their extrapolation strategy from one dose to the other. And I'd love to share with you the Zanubrutinib story. Then I'll begin to discussing extrapolation strategy to a different indication. Zanubrutinib was approved as a second generation PTK inhibitor. BTK, also known as Bruton kinase inhibitor, plays a critical role in B-cell receptor signal pathway. So the dysregulation of this pathway has been observed in multiple B-cell malignancies. Ibrutinib, the success of Ibrutinib is certainly the fairy tale story in our industry. So in 2013, Ibrutinib became the first approved BTK inhibitor by FDA as a breakthrough therapy for patients with mantle cell lymphoma, MCL. Now, let's look at, So after Ibrutinib approval, BTK inhibitors are transforming the treatment paradigm in the hematological malignancies. The second generation BTK inhibitors, such as Acalabrutinib and Zanubrutinib, are designed to enhance its selectivity. So similar to Ibrutinib, Acalabrutinib and Zanubrutinib are covalent inhibitors. And currently, the dose regimen approved for a BTK inhibitor can be given either once a day or twice per day. Now, let me take you back to the beginning of the story. So Zanubrutinib entered clinical trial back in 2014. And a serious dose ranging from 40 milligram all the way to 320 milligram were studied in the dose escalation cohorts. And subsequently, based on PK/PD, initial safety and efficacy, 320 milligram QD or 160 milligram BID dose were selected to evaluate in the dose expansion cohorts. At the time, the data showed comparable efficacy and safety between the QD and BID doses. So which one should we take it forward to the pivotal study? Now let's take it back to look at the clinical PK data. So what you're looking here is on the right. So on your right is time concentration PK profiles following QD and BID doses. Since the PK is linear, we see comparable AUC between the two regimens. Now, looking on the left is the free drug concentration profiles with the two different regimens, QD and BID. And these free drug concentration were compared to the IC50 or BTK inhibition. So what you can see here, even with the QD dose, the C-trough concentration over IC50 of the, with ratio of 2 is much higher than 1/8 that has previously observed for ibrutinib. So consistent with this sustained PK exposure, we observe sustained and deep BTK inhibitions. So in the middle, what you can see is there is a sustained BTK occupancy or inhibition in the PBMC samples. Now, on your far right, what you can see is the BTK occupancy in target tissue of lymph nodes. We observe more consistent BTK occupancy with the BID dose compared to the QD dose. The median occupancy for the QD for the BID dose is 100% compared to only 94% for the QD dose. So at that time, we actually did not know whether those difference between the BTK occupancy can translate into clinical meaningful endpoints. However, in order to maximize the success of the pivotal study, our team decided to take the BID dose forward. And so the BID dose was studied in the phase 2 MCO study, as well as several other pivotal studies, including pivotal study for Waldenström and 2 pivotal studies for CLL. Now, consistent with this PK/PD hypothesis, we actually did observe high response rate in patients with MCL as well as many other B-cell malignancies. And in particular, we observed very high ORR and CR rate in patients with MCL, and these certainly formed the established foundation for our first NDA application for accelerated approval for the MCL indication. So what about the QD dose? So at that time, obviously since BID dose was studied in the pivotal study, the BID dose was recommended as our label dose. But at that time, encouraged by our FDA colleagues, the project team also explored the potential for a QD dose as a label dose. But how do we kind of fill the gap? As Mark mentioned earlier, the MCO is orphan disease indication, so very much constrained by the small number of populations. So as you can see here, that we, in the first one, phase 1 study, we observed comparable response rate between QD and BID doses. However, the sample size was quite limited. But importantly, we observed comparable response rate between the QD and BID doses for several other B-cell malignancies, including CLL and Waldenström's. So that more or less support our initial MCA approval. Now, let's take a look at other totality evidence that we have to support the QD dose. We conducted exposure response analysis for ORR endpoints. And as you can see here, we did not observe apparent ER relationship in ORR in patients with MCL. Again, here is the ER analysis for the AE and safety endpoints across patients with various B-cell malignancies. So I'm going to point it out that the QD regimen essentially only differ in Cmin and Cmax. So you can see here, really there are no relationship between those PK parameters and safety endpoint of interest. So essentially, the ER analysis, along with other totality evidence, was used to support approval of both BID and QD dose, even though the QD dose was not studied in our pivotal trials. Now, since that we are able to receive approval of both QD and BID dose for other indication as well, including Waldenström, MCL, and also more recently CLL indication. We do recognize the limited clinical data we have for the QD dose. As such, our team continued to collect additional efficacy data post-approval. And as of today, we have collected more than 2,016 patients, those with the QD dose. And the data continue to support the label dose. We believe that once-a-day dose is a more patient-centric dose regimen, since MCL and the other B-cell malignancy affects mostly elderly populations. With the importance of post-approval data collection as well as the analysis, I would like to give you two other examples. The first example is to use ER analysis to bridge to a different dose. And this example is Dosages-Session, it's a TKI for CML. The initial approved dose was 70 milligram BID, and the sponsor conducted additional ER analysis, and based on the ER analysis and post-approval data collection, the label was changed to 100 milligram QD. Here is another example where this is for a PAR inhibitor. Initial approved dose was 300 milligram QD, but however, the majority of patients has dose reduction. So the sponsor conducted additional ER analysis and identified patient with body weight less than 77 kilogram or the baseline platelet count are two significant predictors for developing AE. And based on the ER analysis and also additional data collection post-approval, the label was changed to 200 milligram QD. Okay. Now I would like to switch gears to talk about the selection, dose selection for a new indication. And I would like to use BCL2 inhibitor as a case study. I know I see colleague in the audience, a colleague from Genentech and AbbVie, so I'm definitely looking forward to additional discussion. So here, some of you know that BCL2 family proteins play an important role in the apoptosis pathway, and Venetoclax is the first-in-class BCL2 inhibitor. Venetoclax approved dose for CLL is 400 milligram. And various different ramp-up schedule is necessary to mitigate potentially life-threatening tumor-like syndrome. From 400 milligram for CLL to other indications, I would say are not straightforward. For example, let's look at AML, for example. Multiple combination regimens were studied during clinical development, and the approved dose for AML now is 400 milligram for Venetoclax. However, real-world data showed that patients with shorter duration of treatments tend to have either comparable or longer overall survival. That's not looking down in multiple myeloma. And so in the phase 1b study, it was determined 800 milligram was the dose to move forward in the pivotal study. Combination was bortezomib and dexamethasone with Venetoclax. However, the phase 3 study showed that this regimen or dose may have detrimental effects on overall survival, except for that subset, the patient carrying on specific translocation 1114. So using this example, we know that the CL dose to other indications are not straightforward and require additional optimization efforts. So those are the three areas that will certainly require additional attention and optimization. The good news is, as an industry, we have come a long way. Now we have a collection of MIDD approaches to help to address those specific area of optimization. For example, in our organization, we have developed a QSE model to help the team to identify different ramp-up schedule. And we also employ myelosuppression model to help the team to identify different dose schedules. Let me just really quickly share you how we use myelosuppression model to support our team. So this is one example, although it's not BTK inhibitor specific. In this particular example, one kinase inhibitor needs to be added on top of another kinase inhibitor. So the question the team was asking, what is the optimal dosing schedule to minimize the overlapping toxicity? So for that, the team developed a myelosuppression model based on original work by Faber et al. So this shows a pretty good agreement between predicted and observed peripheral platelet counts. And based on the model that we are able to conduct various simulations, that I want to really draw your attention to that red arrows there. That is the threshold triggering high-grade thrombocytopenia. And based on the simulation, we learned that the dose regimen with low dose 2-week lead-in significantly reduce the risk of high-grade thrombocytopenia. And based on this simulation outcome, we're able to advise our team and put it in a study protocol. And indeed, the model simulation were later confirmed by our clinical results. Now, quickly move on to another application of MIDD. In this particular example is early project combination with other existing therapy for patients with multiple myeloma. The question the team is asking is, what is the recommended dose? And also they want to understand the probability of success compared to the existing therapy on the market. For this, our team developed a TGI OS modeling framework. I know this OS TGI has been championed by many people in our field, especially colleagues from Genentech. And here it shows a very good agreement between predicted and observed and protein data. And for this, we're able to identify significant independent predictor for overall survival in patients with multiple myeloma. Before I wrap up, I just want to point out a couple of my thoughts. You know, as an industry, we have come a long way, yet challenges still lie ahead. And dose optimization, I know most of the time we been primarily focusing on dose. Lately, we have encountered several situations in our team that duration, optimization of treatment duration could be as important. The other thing is, how do we maximize ER analysis based on different treatment duration study in the program? Yes. The other thing is about the the ability for us to predict long-term outcome, such as overall survival. I'm sure many of you have heard the news with multiple recent ODAC. OS endpoint is critical in any drug approval. But the question we want to ask ourselves is, how do we use MIDD to be able to predict OS endpoints? I know I myself know building such a predictive platform for OS is a very audacious goal that requires a large data set and enterprise approach. Perhaps this is the opportunity for industry-wide collaboration and FDA leadership. Okay. So I would just love to wrap up. Then today I have shared with you the Zanubrutinib story, the ER analysis, along with other MIDD and totality approach was used to support approval of the 320 milligram QD dose that was not initially studied in our pivotal studies. So in terms of extrapolations to a different indications, we now have a collection of MIDD tool to help us to conduct additional optimization based on the initial indication. With that, I really love to thank you for your attention, but we want to thank the Zanubrutinib Global Fighting team. Also want to thank our patients for entrusting us. So this is the inspiration I want to share with you. These two gentlemen met in our clinical trial in our first in human study, and they told us the side effect profile of Zanubrutinib is wanted to enjoy life more and want to travel more. So they met and they made the t-shirt together and travel around the world. And with that, I really want to thank everyone for your attention. Thank you.
Thank you, Dr. O. I think that nicely rounded out the session with considerations for targeted oral agents. And with that, I'd like to ask Dr. Stacy Shore to come to the stage, who will moderate the session and also briefly introduce our two additional panelists.
Good afternoon, everyone, and thank you for still being with us. I want to go ahead and introduce our two additional panelists, both from the FDA. First, Dr. Mark Thierry. He is a medical oncologist and the Deputy Director of the Oncology Center for Excellence, and as well as Raj Madhabushi, who is a pharmacometrician by trade, but is working in our guidance and policy team within the immediate office in the Office of Clinical Pharmacology. So welcome, both of you. And with that, I want to go ahead and kick off the panel discussion. We heard a lot of very interesting discussions on different modeling-based approaches that can be used to navigate from one tumor type or one indication to another indication. And one of the things that has struck me, being a pediatric clinical pharmacologist in some degree, is that when we look at extrapolation for pediatrics, we think about similarities and differences in disease. And from that continuum, we decide whether or not we can borrow that dose from pediatrics to adults. And I see a lot of parallelism here because we have to look at all of these different factors. And so I guess one of my questions to the panelists is, what some of the factors that we need to think about when we're looking at similarities versus differences to figuring out, can we carry the same dose forward, or do we need to look at different dosages?
So maybe I can say a few words. Actually, I fully agree this pediatric extrapolation framework may potentially apply to the subsequent indication. Actually, pediatric is part of the subsequent indication. So I think the difference is maybe because pediatric, they also have PK difference. You need to do that allometric scaling down by either by enzyme or by PK. There's an ontology expression for the metabolic enzyme. I think for the newer indication, besides PK, in a certain situation, we do have PK difference, but majority will be the biology difference. So you basically compare whether the biology is different. You can either leverage preclinical data or clinical biomarker data to demonstrate similarities or difference. And then based on that, we will do the decisions. I agree. I think I really love to, I'm planning to read the pediatric extrapolation framework to further leverage that. I think that's a really good thought. Thank you.
Thank you. Raj?
In general, I think there are some features which from the pediatric extrapolation framework that could be borrowed. I think there were some points that Dr. Retain brought up in his presentation, which could be useful for setting up a framework here. The tissue of origin, the tumor burden, and molecular genetics, I think those are definitely key factors which can be taken into account. And in fact, I think we have applied going into pediatrics already using that kind of an approach. See, fundamentally here, it's not whether the drug is effective or not. That has been demonstrated with the first approval, right? It has demonstrated. The point is whether it would be effective in an indication which is different from the first one. So the question is, how proximal is this indication to the first one that has been approved? What might be the other features with respect to the tumor itself or the oncologic condition, as well as how would the drug behave, right? These are the 2 points that Dr. Retain brought up. I think there are some features which could be useful to actually help set up how to systematically go into other indications and thereby be more efficient, as there might be situations where there could be abbreviated clinical development programs where it is appropriate. In other situations, you might have to do a more in-depth evaluation, but this framework could provide some hints into which of the 2 pathways might be more amenable given setting and what are the uncertainties that exist.
Yeah, just want to add, it's definitely an interesting question, very thought-provoking question. I'm actually, my first reaction is to ask this question back to the FDA. Will FDA actually accept a subsequent indication based on extrapolation principle similar to pediatrics without a further dose optimization, right? So obviously, the extrapolation principle for pediatric is based on pediatric being a vulnerable population, right? There's some wiggle room for that vulnerable population, but it is a very interesting thought, right? Because if we can demonstrate the biology of the differences in disease, the treatment, we could potentially demonstrate the response to treatment is similar, or how is it being different, through in silico approach, through QSP approach, and PK differences is easy to demonstrate and easy to predict, right? So if you can do all this, is extrapolation principle all there is for certain cases to enable a subsequent indication without further dose orientation. So I think that's a very interesting thought. Looks like from FDA there is acceptance, at least in concept. So I'm glad to hear that.
Thank you. I want to go ahead and invite the audience up to the microphones if you have any questions.
While hi, Mark Thierry, FDA. So, while the audience is coming up to the microphone, I just wanted to, just say, I had a couple thoughts, a few thoughts I just wanted to share. I was fortunate to be able to spend the day here and heard some excellent presentations regarding, a lot of the scientific and clinical considerations regarding, thinking about combinations and how to extrapolate dosing and how to, not extrapolate dosing, how to think about dosing for combinatorial regimens. But also now, this subject of this session, I thought excellent, presentations and discussions regarding scientific considerations for thinking about, dosing, for subsequent indications. And, initially I thought about, going through some of these scientific considerations, but I believe, our presenters and panelists really, I think, brought out fantastic examples, fantastic, considerations. So I just wanted to share, 3 kind of thoughts, regarding this concept in general. So first, and actually I have to give credit to Dr. Pazzer, who's my boss, for the first 2. And I'll just use two quotes, which he has used before, and I think really get to the heart of, this issue of thinking about dose optimization. One is that, no one has the right to subject patients to excessive toxicity. Okay, so, thinking about Project Optimus and how this came to be, certainly the scientific advances, and all the investments that we've had, over the past decades in cancer research, basic translational, has resulted in certainly more therapies and more effective therapies. A major impetus for Project Optimus was calls from patients to not only have more therapies, more effective therapies, but more tolerability of the therapies, and not thinking about development of drugs as they are, were traditionally developed in the cytotoxic therapeutic paradigm. Patients are on drugs longer, with molecularly targeted therapies, immunotherapies often. Toxicities do build up and do impact, patients' quality of life and consideration of what a tolerable dose is. One of the other, principles I think really just cut to the chase, for a lot of the discussion today, what Rick likes to say is that dose is the foundation of drug development, right? So having the wrong dose is like building a foundation on quicksand. And I think a lot of that really comes through in these discussions. And you've heard today, I think, a lot of discussion and emphasis, for better characterization earlier on in drug development, and the fruits of those better characterizations coming through in thinking about combinatorial development, as well as, thinking about subsequent indications. And then the third just concept, and I'll just finish up with this, is benefit risk. I think there was, earlier, question regarding kind of thinking about benefit risk in different, indications and settings. And that is a major consideration from a clinical perspective, this benefit risk. We have obviously a span of treatments for patients with cancer, or patients who had previously had cancer, in terms of thinking about definitive therapies, or patients who do not have cancer, but maybe at high risk, or healthy people who have a risk of developing cancer. So thinking about chemo prevention and tolerance for adverse events and toxicity is certainly much different for what patients may consider tolerable in the late line setting. And I think as an example of kind of the rush to get novel therapies, promising therapies to patients, we have had this paradigm of single arm trials in a very treatment refractory setting. Okay, and very difficult to really understand toxicities in that single arm trial, although we've had kind of sufficient information to think about a positive benefit risk in discussion with patient advisory committees, and thinking about the benefit risk framework that we have. But I do think it gives us all pause regarding this paradigm, when we do see multiple different therapeutic classes coming to light with randomized controlled trials now that have demonstrated potential detriments in overall survival. And it's, very apparent that there could be toxicity considerations that are present in those development programs based on poor, or inadequate dose, optimization that had occurred early on. So the focus of Project Optimus at the outset was really trying to change that paradigm and have better pre-market characterization, more focus on pre-market characterization of a dose or range of doses. However, still with that principle, overarching principle of expeditiously and efficiently getting novel therapies, promising therapies, into the hands of patients. And just the last point, I actually thought of to your question and look over to the FDA. I was told not to make policy from the podium. Okay, so just realize that that we're providing and sharing our experiences, but not necessarily or not providing making policy from the podium. And for individual questions, we would not have you ask about specific products. Obviously, these are general principles. So.
Hi, I'm Helen Moore. I'm at the University of Florida and was very happy to see lots of people presenting extensive modeling used in determining doses. So my question is, we've seen a lot of methods that use simulation. So we'll have a nice model, it's well validated, and then a lot of different simulations of regimens and picking one that seems good out of all of those. But we have techniques, if we have such models, we can actually optimize. You can determine your goal. You know, this is our measure of efficacy. This is our measure of toxicity. We can optimize both of them, or we can use relative weights on them. So these methods exist. Other fields use them extensively. Anywhere there's something that's big at stake. So aerospace, automotive, oil and gas, manufacturing, supply chain management, even manufacturing in drug development, we are using optimization methods at our plants, but not in our drug development programs because we in the past haven't had models that we really had as much confidence in. So if we do have confidence in these models, and that may be the one question, but if we have models of neutropenia, models of insulin glucose, models of viral dynamics that we use for developing antivirals to new pandemic viruses, if we have such models, and we have such optimization methods, I'm curious, maybe perhaps first from FDA, what is your recommendation for, based on your experience, how do new applications, not new methods, these methods are old, how do these new applications become accepted? What is your recommendation for the evidence needed to be able to make a submission, or if it's on the industry's shoulders to do that, then what is your encouragement to industry? And perhaps industry wants to also speak to this.
Yeah, but so if I were to summarize, your question is if there have been advances with respect to some of these quantitative approaches, they have been applied in other areas, and if they are mature, what does it take for them to be applied, right? That's the question in general. I think the general answer to that is the models should be fit for purpose. They should show the kind of credibility that is required for the question at hand. And we have to have a very good understanding of what are the limitations of the model also, because especially if we are thinking of applying, so there are two aspects. If we are trying to apply models to inform our trial designs, that's one space. If they are being used in lieu of actually generating data, clinical data, the bar would be very high. There has to be a high level of precedence and confidence that would need to be generated. And it also depends on the settings. So what we are trying to encourage, at least from the Office of Clinical Pharmacology here, with respect to be able to make progress towards the direction where we can all collectively have that kind of confidence with models in certain settings, is to use this concept called as a risk-based or risk-informed credibility assessment framework, to provide that kind of use that kind of a framework to walk through the various aspects in that particular framework to be able to help identify what is the kind of evidence that is required to if I can use the word, qualify or validate these models, and then ask the questions that are remaining is, what is the model influence and what is the decision consequence? Consequence of making a wrong decision, what does it lead to? And based on that, one may be able to not only set up how to go about these modeling exercises, but also see whether something is feasible or compatible in a given setting. And it becomes a very highly case-by-case kind of situation. But there is a general framework that could be leveraged to be able to make these progressive steps towards a direction where there are situations. We use PK-based models in certain settings for dosing regimen, right? There are well-established situations where we have gone from once weekly to twice weekly dosing situations. So it all depends upon what kind of maturity we get for different kinds of applications. But this risk-informed credibility assessment framework should provide this kind of a broader framework and vocabulary for all of us to actually try to figure out what are the places where we have to make these progress. and gain collectively that confidence. It's not just confidence of a technical expert or a modeler. It has to be all the decision makers who have to be able to have that kind of an understanding of what is the putative value of these models and what are the uncertainties, such that we can make informed decisions.
That's helpful. To clarify, I'm more interested in, we have lots of simulations we run of different dose regimens. So instead of running 10 different regimens and looking and finding out what the predictions are for the outcomes, we could run an optimization. We're able to do that on the types of models we have now. And then that's what we would test in the clinic or preclinically. So it would still be validated some way, but just to reduce the number of large simulations of virtual patient populations, just to actually use an optimization method instead.
No, I think that would be a step in the right direction to be able to more efficiently get to translating some of these efforts with respect to applying these quantitative approaches to be able to inform your study designs. That probably would be a good idea, I would think.
Yeah, can I add? I think that's a very good question, Helen. I think I just reflecting, what we have been talking about this afternoon. I think we did ignore one important aspect. We're talking about dose optimization for subsequent indications, right? How am I going about this randomized, dose findings? But the first step is actually to have the POC, proof of concept. Right? I think Dr. Retain in his remark did talk about this, right? So if you have prior dose optimization when it comes to subsequent indications, right? The first thing you need to think about is your subsequent indications, how different it is from, in what aspect is different from your initial indication. And from that aspect will inform you, what confidence you have in your model, right? So you could have model that you have fairly high confidence in this initial indication, but whatever you predict in terms of differences, does it have any impact in the subsequent indication, right? Based on the subsequent indication specific characteristic in disease, right? So I think the first step is POC, right? Once you establish POC, you know what type of confidence you may have in terms of modeling approach. Then again, if you do have, fairly strong evidence in the disease is fairly similar, then I think a model-based approach could be, completely acceptable, right?
Thank you. My name is Malaz Abitarf. I'm from Daiichi Sankyo, the company that developed Enhertu, and I'm glad to see some of the data being presented from our competitors saying that we did the right thing. So thank you. My question is probably very limited, very related to what Helen mentioned. I mean, I could tell you that we had a lot more data than what was presented, and we had a lot more studies and dose regimens that we looked at to say, we want to understand the therapeutic window for this drug. We want to understand the range of exposure response analyses and what is our tolerance. And there's a lot of discussions. We quantify the risk, we quantify the benefit, and then there's the judgment of how do you weigh how much risk you're willing to take versus the benefit. And you do all of that work. So we did all of that for these compounds, and then you do it for the next ADC that has very similar components to this, and then you do it for the next ADC that has very similar components to it. What is the panel's reaction to what we can use from one compound that has some differences and some similarities to another? We have similar payload. We understand the relationship between the payload and potential hematological toxicities. I have pretty high confidence of that relationship. And I really don't need to do a lot of dose ranging for that new ADC for that specific reason. There could be other reasons why I need to do it, but for that specific reasons, I don't. So what is the panel's reaction to this from that perspective? Thank you.
Yeah, I think that's a really good point. Actually, I do feel it's quite important to leverage platform learning for the ADC with the same payload. Actually, that's what we did for VCMME ADC. We found out actually all the VCMME ADC, regardless the target, they all end up with similar MTDs. So I think sometimes the the unconjugate payload, the payload actually drive the tolerability. So I think the learning really helps. Actually, we combine all the data together to build a matter pop PK analysis and also to understand the peripheral neuropathy, the common AE across the ADC. So I do feel there's a value. Again, you need to put the uniqueness and also commonality together and bring that specific from the platform learning to your molecule. So I think this is definitely, I feel there will be value if we have multiple ADC in the portfolio. Similarly, for T-cell engager, there is a common AE, so you can leverage the learning, for example, step-up dosing, and also even leverage the modeling, the structures to apply across different platforms, but you just need to tweak the target, etc.
Well, I guess, you know, I would say you have a reasonable hypothesis, which is the dose response, both efficacy and safety, are going to look very similar to your previously well-studied compound, but it's only a hypothesis. And so you have to do studies to test that hypothesis. You wouldn't have to start with a blank slate, but you still only have a hypothesis. And there's going to be differences in tumor burden. There's going to be differences, perhaps, you know, if the payload's different in toxicity relationships. So I think it's a reasonable starting point, and you don't have to go threefold higher and threefold lower, but I would still be interested in seeing some evidence of whether or not there's a flat or you know, a dose response relationship.
Exactly. Thank you. I did not want to repeat what Helen mentioned, that yes, this is not in lieu of not doing anything. It's just going with the hypothesis that it is flat. I'm going to do one dose level, maybe a little bit less with a lower dose level, and then demonstrate that the relationship, it doesn't have to be flat, but it's very similar to what I think it is. So it's different than going with, I'm going to look for something to make me believe that I'm not right, versus I'm going and I need to show the same thing with the same level of robustness and the sample size and all of that. Thank you.
Yeah, I think the model help you nail down the different options, but not totally replace your clinical trials.
Yep. And I don't think we're there yet into replacing clinical trials completely, but again, there's a difference between going with, here's what I think it is going to do. I'm going to do one dose and demonstrate that it is similar to what I think it was going to be, versus having to do the full range again, similar to previous trials.
Okay. So we're going to take a question from online chat, and this is a general question, but perhaps we could spin it in the new indications area. So could the presenters please comment on the use of receptor occupancy during dose optimization? In many cases, tumor RO data is not available, and peripheral RO data cannot represent the level of target engagement at the site of action. It's for anybody who'll take it.
Well, you know, I think it depends what the preclinical data shows to how well receptor occupancy correlates with efficacy. If you have that data, then you've got a biomarker that is reasonably likely to predict efficacy. If you don't have that data, you're shooting in the dark.
Yeah, I also want to briefly comment on receptor occupancy, because this is a key clean farm concept, right? So if you're theoretically, if you fully saturate your tumor RO, then you don't need to push higher dose. So you won't gain additional efficacy benefit. But the challenge for peripheral RO may not necessarily reflect, you know, a tumor RO. So I think we make a lot of hypotheses, you know, build a model to build that RO. So there's still uncertainty. So so that's the reason you still need to collect clinical data to really finding your dose. But those RO calculation at least can serve as a minimal bar, because you need to saturate peripheral RO first. If you could not do that, then you know something wrong with your drug. So I think the RO is somewhat useful, but you need to put into context your mechanism actions. For example, for antagonists, you need to maximize the RO. But for antagonists, you don't need, right? But the RO, for example, the example I'm showing here is actually help us to dissect the confounding factors to really help us understand the ER relationship. But we need to put the RO concept in the context of mechanism action and biology.
Yeah, I just want to add, I agree. I think tumor RO is extremely important actually from a early development standpoint. So I recall yesterday there was a session talking about tumor heterogeneity, right? Obviously, even in the scenario that you do collect tumor biopsy and measure tumor drug concentration, tumor RO, there could be a lot of pitfalls with that methodology, right? So I think in the industry, we're really trying to leverage QSP, and these models becoming more and more sophisticated to help us understand the tumor RO. And the other advantage of tumor RO is really, as I presented, it can help you evaluate different potential scenarios, right? Let's say if you have certain understanding of a particular tumor biology, right, in terms of tumor load, in terms of target expression, you can put that into the mechanistic modeling for tumor RO to potentially guide you with the dose optimization for a subsequent indication.
Azhar Hussain from Merck. Just want to follow up on the comment and question which Malaz has just asked regarding building upon the experience, what you gain over a period of time, specifically with respect to ADCs. My question to the panel is perhaps two questions. The first question is, once we sort of established exposure-response relationship in a particular tumor type, would we expect to see a similar behavior across other tumor types? From theoretical point of view, you would expect that the slope is going to be similar, but the magnitude of those relationships could be different because the response rates could be different. The second question, perhaps if I could, is, once usually we develop the exposure response in the late line, right? That's the first indication we go for. And these are the patients who have been heavily pretreated. Their benefit risk profile could be slightly different. Their toxicity profiles could be different. The response rates could be different. Any experience with seeing whether the benefit risk profile changes as we move to the earlier lines, or in a settings like neoadjuvant or adjuvant settings where the benefit risk may be slightly different? Thanks.
Maybe I can address the first one. So what's the first question? I kind of forgot.
Yeah. I mean, whether the response relationship, is preserved across tumor types.
Yeah, actually, I don't feel you can automatically assume the ER relationship will be the same across indication, because I think that it will depend on the target expression on your specific tumor. Because in general, some of the target, they may have similar level of expression in different tumors. So in that case, I think you probably can assume there's somewhat will be similar. And I agree with you, maybe more like EC50 versus ED50 versus the Emax, because different disease will be slightly different. But some of the target maybe have different expression on your tumor. So you need to leverage your preclinical understanding to look into how much target expression and how that corresponding to the tumor cell killing curve, etc., to really understand whether you will think that you will believe the similar dose response curve will apply across indications. Does that make sense?
Yeah, thanks.
And related to the early disease, later line to early line disease, I feel probably the clinician will better comment. But I think from the case, the summary from FDA approved indication, you can see many of when we move to later line to early line, actually they kept the treatment duration. I feel in general in our drug development, when you go to adjuvant space, so you don't want to treatment for patient forever, especially you have chronic toxicities. So I think preferably there's a high expectation to have more tolerable, higher safety and tolerability bar for the earlier disease. Because for this patient, they have curative intent. They need to have relative quality of life to live life a normal patient. So I feel the benefit risk profile will be different. But I think Mark and others will feel free comments.
Well, let me comment. I think if you're treating metastatic cancer and you want to move something from late line to earlier line or vice versa, whatever, I think the dose is the dose. On the other hand, if you're going from metastatic to adjuvant, you may need a higher dose in that setting, because if you're not curing the patient in the adjuvant setting, it's probably not worth giving the drug. And so the first question is your drug potentially curative? And I'd say, like, if it's a kinase inhibitor, for example, it's not. But, you know, if it's an ADC compound, the answer is yes. And so I think that's a more challenging question where you've nicely optimized the dose in the metastatic setting. And maybe you have shown that a higher dose is tolerable but no more effective in the metastatic setting. Could you justify using that higher dose in the adjuvant setting? Perhaps. And I think that would be an interesting regulatory discussion, because you have theoretical reasons why you want to maximize the dose, and you know that dose is tolerable.
And I think we'll actually be able to have more informed discussions, you know, moving forward, as opposed to, you know, kind of the historically we've been, you know, utilizing and relying upon, you know, the one dose that was typically found to be, you know, found at the MTD and taken forward and throughout the registration, you know, trials. And then, you know, putting that in the context of the benefit risk for particular patient populations, you know, as they, you know, are in various contexts of their disease. So I think we'll be able to have much more knowledge of the data and the relationships moving forward with, you know, enhanced emphasis on characterization. And then, of course, the goals of, you know, therapy absolutely impact, you know, the considerations, you know, for, you know, requiring particular doses. Also, in different settings, we may see, you know, patient populations that are, you know, healthier, that may be subjected to higher amounts of toxicity with similar doses, depending on immunotherapy or whatever that, you know, mechanism of action, you know, might be, in the particular mechanism of action. So, you know, having a better understanding of that, you know, will be helpful. And understanding the potential curative potential, you know, of a particular therapy is extremely important when thinking about, you know, how much toxicity is, you know, acceptable. You know, it's a much different, you know, discussion with patients, you know, if there is, you know, curative potential of adding that, you know, regimen versus, you know, considerations at the outset that, you know, maybe just further delaying, you know, progression, you know, as an example.
So I would love to comment on model confidence and model refinement. And then a couple audience mentioned that, you know, typically what we do in the industry is each company use their own data and refine and build a model and validate in a small scale. Recently, our genetic colleagues published a really nice paper based on the TGI-OS modeling framework, and they were able to use the model prospectively predict the OS endpoint. So I guess my question to FDA is there any possibility like we industry can collaborate like together and eventually we can actually build a model for predicting OS using a larger data sets? I mean, I know this, I'm a dreamer, so apologize if this question is out of scope. Thank you.
Well, I think that the quick answer is yes, people can collaborate, and we encourage that in many settings. We often aren't taken up on that call for, you know, collaboration for particular projects, but you know, in terms of the particular situation, and I think you know, the details are very important. You know, and but no, there's absolutely opportunities for collaboration and, you know, think about ways to generate data, collect data, and discuss data with the agency for utilization as a drug development tool in particular settings. So I'll start with that. I don't know if you have additional comments or.
Well, I would just say that Roche would be unlikely to collaborate with you. They just buy you.
Well, the only thing I would like to use this opportunity to make a plug is that we do have this pathway of fit for purpose initiative, which is for developing tools to advance the development in areas. So if there are quantitative tools that can help with moving the needle in a particular area, definitely there is a pathway for having some kind of an engagement to have some qualified tools that will now be accessible for people to make that much more informed decisions for designing their trials and their development.
Just want to have a quick comment. I really like this panel discussion because I was thinking, well, we're discussing most likely, these quantitative methodologies we're talking about, trying to predict, OS, trying to predict safety when we're moving, different subsequent indications or from different lines of therapy. But I think there was very good comment just now from the panelist that you also have to consider the different risk tolerance or risk profile, right? Let's say, moving to an early line or adjuvant setting, you could have a curative intent, then risk profile could be entirely different. It just remind me of earlier today, there was one presentation basically, trying to say that we could use potentially utility score type of endpoint, right, to help contextualize the different doses in different disease settings and lines of therapy.
Another question online. Could the panelists comment on optimizing the number of doses of agonist programs like TLR or STING, where continuous dosing may not be needed but limited to just a few cycles? We may have stumped the panel. Yeah, I will. I think it goes to, these new therapies where we may just have a few doses that we're optimizing. So could the panelists comment on optimizing number of doses of agonist programs like TLR or STING, where continuous dosing may not be needed, but we just have a few cycles? How do we optimize the dose in that case?
I mean, schedule is just as much a part of dose optimization. So the same principles seem to apply.
Yeah, I think I don't have much experience on this specific pathway, but I feel if you understand the fundamental pharmacology, you know that you have memory T cells or other memory system, you don't need that many dose. I think you can leverage the pharmacology understanding to rationalize why you don't need continuous dosing. So I do feel it is for certain level, certain molecule maybe makes sense.
All right, so another question from the online. What are the panelists' thoughts on emerging biomarkers such as spatial distribution of immune cells for IO programs where exposure, response, or receptor occupancy may not be very informative to inform doses?
Can you repeat the question? Sorry, just want to make sure I understand.
What are the panelists' thoughts on emerging biomarkers such as spatial distribution of immune cells for IO programs where exposure, response, or receptor occupancy may not be very informative to inform doses?
I'm actually not very familiar with that particular biomarker, but I can comment in general. I do think biomarker is important. You can consider tumor receptor occupancy as a kind of biomarker, but I think utilizing biomarker, you also have to be careful, in terms of collecting biomarkers, right? So I think there was a discussion yesterday, again, the heterogeneity in patient population. If you collect your samples in, let's say, in a particular, satellite tumors, different tumors, right, within the same patient may be responding, may not be responding, right? So you just have to be careful in interpretation, but I think overall will be very helpful in guiding the drug development for the IO therapy and for, I think, a range of therapies.
I agree as well, but I think biomarker is kind of set. This biomarker you mentioned is more like get your POM probe mechanism. Your molecule work through your pathway, but doesn't provide you the POC because ultimately you need to see the anti-tumor activity. So therefore, there's a caution. I think you need to leverage a biomarker for certain level decision. So for example, as a minimum bar, but should not be the ultimately use that as a decision point for your dose. Because if ultimately you need to achieve POC, anti-tumor activity bring the benefit. So I think there's a fine balance. If you're confident with your biomarker is clinical relevant in the context safety efficacy, you should fully leverage that for the dose decision. But if you don't have that confidence, you can guide as supporting evidence, but should not be used as only determination for your dose without the context safety efficacy.
Thank you. One last question. Can post-approval information on safety and effectiveness be considered just like dosages to be evaluated in clinical trials for a new indication? So basically, how can we leverage information we already have that may not have been collected during a clinical trial, but as experience is gained with the drug?
Well, I'll try. I mean, it just goes to the knowledge of the investigator. And, I think that science is science. I think if you don't understand science, you should not be developing drugs. I'll just start with that. And if you don't understand pharmacology, clinical pharmacology, quite frankly, you shouldn't be developing drugs. And so I think we just need our investigators to up the game and the sponsors to up their game, and we'll all live happily ever after.
Thank you. Yeah, so I'm going to make a quick comment in terms of post-approval data. Just like the example I shared in my talk for Zanubrutinib or even Venetoclax for AML, the post-approval data are just as important. So definitely encourage all of us to not drop the ball, continue to collect the data and additional analysis as needed. But what I see a potential opportunity is the real world data. There's tons of real world data out there, but the challenge is how do we capture the right parameter so that we can do an informed analysis. I think that's something our organization is struggling with because the come in with different data sets. So perhaps I would love to hear more thoughts on this part.
So, from the agency perspective, the dose optimization or the life cycle of the drug certainly doesn't end with approval, and we are always learning more. I mean, the safety databases, particularly for drugs that are approved for, in oncology, are sufficient, but on the smaller side often compared to other therapeutic areas. So there's an expectation that the post-market system is going to be very important for informing further about how a drug is to be used and other information. So certainly other information that will come from additional trials and looking at other doses. I think sponsors and are going to be extremely interested in further evaluating the benefit risk of their drug in different indications, potentially in different schedules. That was brought up earlier regarding the dose optimization that's required, and FDA certainly has the authority to require dose optimization in the post-market if not adequately characterized in the pre-market setting, and there is what we call FDAA-related kind of authority to evaluate a serious risk or identify a potentially serious risk with that. So that's where that authority. So ideally, further characterization in the pre-market is going to, not only be more, efficient, but also lead to less, requirements at the time of, deciding whether the benefit risk is favorable for a approval decision. Is that dose, optimized?
Thank you very much. And with that, I'm going to go ahead and close out session 3 and thank all of our panelists for participating today. Today marks the completion of 2 successful workshops co-sponsored by the FDA and ISoP. These workshops highlighted successful examples in which quantitative approaches helped identify dosages to be evaluated in clinical trials for oncology drug products. With the first one focused on intrinsic and extrinsic factors, and this one focused on new indications and combination therapies. These workshops have allowed us to talk about resetting our expectations for dosage selection and optimization with emphasis on using the clinical pharmacology toolbox to evaluate all available data to identify a recommended dosage. We've covered a lot of material today. In the morning, we started off focused on understanding regulations and impact of response variation. We discussed the non-clinical and clinical considerations for novel combinations and new indications from regulatory perspective. We also learned how modeling can be used to evaluate response variation and in turn support dosage selection for combination therapy. Next, we focused on dosage optimization for combination therapies. Combination therapy was broadly defined to include discussion on 2 or more novel products, 2 or more approved products, or 1 approved and 1 investigational, including drugs with limited or no activity when administered alone. We learned about and discussed novel study designs for combination therapy with substantial benefits compared to traditional dose-finding trials. We learned about model-based approaches to optimize dosing regimens for novel combinations and heard several great examples in which different approaches were implemented to select a dosing regimen for a combination therapy. We'll learn how different modeling may be used to understand the dose response and exposure response for activity and safety, and how these two surfaces can interact, and how these analyses together can help us design clinical trials and optimize dosing. In the afternoon, we touched on new indications or settings, such as different disease states, treatment lines, or oncologic diseases. Several interesting examples in which the approved recommended dosing regimen varied across indications were highlighted, and we discussed the factors that should be considered when optimizing a dosing regimen for a new indication. We discussed the potential impact of limited or no dosage optimization for the initial indication on subsequent indications. And we heard a lot of great examples of model-informed development for antibody-drug conjugates, bispecifics, immune therapies, and kinase inhibitors, with a focus on the quantitative clinical pharmacology and mechanistic modeling. Some of the key takeaways from today include practical and feasible considerations play a role in study design and selection of dosages to be evaluated. Collaboration across multidisciplines is necessary to look at all data, to design the trials and select dosages for evaluation. And models can serve as a complementary tool to support design and studies and select dosages for further evaluation. Collectively, the programs, initiatives, and tools described in the individual talks and in these panel discussions showcase the means and methods to identify an optimum dose for drugs given in combination or to be evaluated for a new indication. But additional conversations are warranted to continue to identify alternative trial designs and analyses and advance other sciences such as biomarker sciences to better inform dosage optimization. FDA looks forward to continue to engage with industry, academia, and many others, especially our patients, caregivers, and advocacy groups, to identify and optimize dosages for all patients before approval, with an understanding that some adjustments will be necessary as our understanding of the drug safety and drug response variability expands in the post-market setting. ISoP and FDA hope you enjoyed attending our event as much as we enjoyed putting it together. Thank you all for attending, and safe travels for those departing home. Good night.